It's my great privilege to welcome you all and talk about one of the topics that's very near and dear to my heart, but I think is also a central priority for Dana-Farber, and that is accelerating drug discovery. And I didn't pick the title for this breakout session, but I think it's a very fitting title for the two people you will hear from. My name is Fischer. I'm the director of the chemical biology program at Dana-Farber and a professor in biological chemistry and molecular pharmacology, and Kathy Wu after me, who's going to speak about a different topic, but all ties together in drug discovery and accelerating drug discovery. I think I just want to spend 1 minute on giving kind of a broad overview. When we talk about accelerating, I think there are various ways we can do that. We can accelerate the process, we can accelerate the way we do clinical development, but I think we've just witnessed one of the greatest examples of acceleration in drug discovery, which is the development of the COVID vaccines, which are really, the reason we could accelerate it so much was that there was a new technology. And I think if you look back at the history of medicine, there have always been these inflection points when new technologies, new advances in therapeutic sciences were introduced that led to great acceleration or really expansion of scope. And I don't want to go through all of these examples, but I think one we can all relate to in the context of cancer treatment is the invention of therapeutic antibodies, now probably 3 decades ago, that really opened up an entire new area of therapies that are now a cornerstone of every treatment regimen. And similarly, in the space of small molecules, we've always had these advances where things changed, and I'm going to walk you through this a little bit. Let me just take one step back, and I think we've just heard about the importance of chemotherapy, and that's the traditional paradigm of treating cancer, where you have these rapidly dividing cells, and the basic principle of chemotherapy is that you have a toxic agent that is just a little more toxic for a cell that's rapidly dividing and can't cope with this additional stress. And that works fantastically well in many cases. We've just heard about the cure rates in pediatric. I have to say that children generally tend to deal with the toxic load a little better than adults, but it comes with side effects, it comes with limitations, and I think especially in the adult space, there's limits to efficacy with some really great exceptions, but it's clear that there are additional tools needed, and it's not necessarily going to replace it everywhere, but there are additional tools needed. And about 20 or 30 years ago, we had one of these steps where we really had an inflection, and that was the beginning of what I would consider really a paradigm shift, the start of targeted therapies. And I'm going to give you the most classical example, which is where the genome break creates a mutant protein called BCR-ABL, which is a kinase that's activated and really is the cause of these cancers. And it was the realization that there are these mutant proteins, and then the creation of molecules that specifically inhibit this kinase in this case. And now you have an agent that not is generally toxic to the body, and the cancer is a little more sensitive, but you have something that's specific to the cancer and that treats specifically the cancer. And we've seen a tremendous progress, and we continue to see new agents, but there are limits, and we always run up against these limits once we really start to exploit technologies. And while a kinase has this wonderful little pocket where you can fit in a small molecule and you can inhibit the activity of this kinase, we've now learned from another corner of really advances, sequencing, big genomics data, we understand the mutational landscape of all our patients, that not all causes or all contributors to cancer and other disease look like this. Many look rather like this. This is a transcription factor. And even without going into any detail of protein structure, you can probably recognize that one is much more disordered. It's a pile of spaghetti, as I would refer to, while the other is a very structured noodle. And one, I can imagine I can fit a small molecule in, but it's very hard to fit something that specifically binds and distinguishes one pile of spaghetti from another pile of spaghetti that sits next to it. And so we need new technologies. We can't really find molecules to bind to that specifically. But what if instead of doing so, we could just dispose of it? And so I'm going to give you one example of a relatively recent advance in therapeutic sciences where we've been able to do this, where we've been able to essentially devise molecules that act like one of these garbage trucks, take the disease-causing pile of spaghetti and throw it out into the bin. And the reason we can do that is because nature is always a step ahead. Nature has usually done what we're trying to do already millions of years ago, and nature has developed or evolved a system that's called the ubiquitin proteasome system, which can specifically target any protein in a cell and dispose of it by shuttling it to a big, what I would call the cellular garbage truck, called the proteasome, and it gets chopped up in little pieces into its amino acid constituents. And so about 20 years ago, people thought about, well, can we hijack the system? Can we make molecules that would tell the cell to take care of, for example, an oncogene product and just send it to the trash? And in fact, we can. And I'm not going to go into too much details here, but we can now make molecules such as the one shown here, which artificially recruit a specific protein to the cellular destruction machinery, lead to ubiquitination and degradation of this protein, and degrade it. And so this is great conceptually, and it's been around for 20 years, but it hasn't really gone to any therapeutics. And the reason is often because there's something else needed. There's a lot of skepticism, and rightfully so. Before you put something in a human, you need to cross a lot of bars, there are concerns, and when this first came up as an idea, I was back at Novartis, and people would tell me this is impossible, right? You can't do it, you can't just put a molecule in a patient that would degrade a protein, it has some toxicities. It turns out what you sometimes need is a lot of luck. And in this case, luck came from a very old molecule with a very unfortunate history, and that's this molecule on the left, which is called thalidomide. Was used in the 1950s, widely in Europe and some other countries to treat morning sickness, and unfortunately carried severe embryo toxicity, and so many children were born with severe birth defects. Obviously, it was withdrawn rapidly from the market. It's noteworthy it was never approved in the US, mostly due to the work of one woman at the FDA, Frances Kelsey, but it was rediscovered in the late 1990s to have activity against multiple myeloma and some other hematologic malignancies. And people didn't know why, but they started using it. And so now you had thousands of patients being treated with this, and it turned out to be highly efficacious in multiple myeloma. And 10 years later, several groups, several of those at Dana-Farber, including Ben Ebert, Bill Kalin, I was still back then in Switzerland, but we were all working on this problem, why do these molecules work? And it turns out it works exactly as this idea. It uses the ubiquitin proteasome system to degrade two transcription factors, Icarus and Aiolos, that are those piles of spaghetti in a cell that are causative and for drivers of multiple myeloma, and degrading those two proteins can lead to curative outcomes. And so, well, not curative, but improved disease management. And so now you put the idea together with a serendipitous discovery of clinical proof of concept, and you get a real acceleration in a field, because now suddenly people will believe you that this can actually be done. It really expands the scope, because if we look at what classical inhibitors can do, it's a small sliver of the entire proteome, but drug targets are across the entire space. And so now it's not that targeted protein degradation can cover all of this, but I think the concept here is that expansion of therapeutic modalities, similar to what we've seen with antibodies, can really expand what we can do therapeutically. So how do we play into this? It's a complex problem, and this is just ignore the details here, but there are many parameters that we don't understand. It's a very novel concept, and when I came to Dana-Farber, I really set out to think about how can we make this accessible? Because if we make it accessible, people will play with it and people will understand what can be done with it. And so we raised three main questions. One was, can we understand the targets that are amenable to this? Because it was obvious with antibodies, which are extracellular, and everything that's on the surface on a cell, you can target with an antibody. Everything that's within a cell, you can't. In this case, it wasn't that clear, and we developed tools to do so. Can we understand some of the principles, and can we make them widely available? And then we will have biopharma and academic centers pick them up and make new therapies. And can we precedent some targets that are either hard or have other hurdles, and again, encourage others or do it ourselves to develop new therapeutics. I'm just going to give you very, very brief examples. One way we went about mapping or making targets tractable was that we just generally mapped the landscape. And this is a great example where we see just convergence of technologies, which is something else that's happening right now, and where we have high throughput genomics, proteomics technologies, we have better approaches to computational approaches, we have machine learning and other things that make large-scale initiatives possible. And it really boils down to we can learn some of the grammar that underlies degradability. So we can learn the grammar that defines whether a target is tractable to this approach or not. And we've done this over the last couple of years, and I think the best way I look at impact is not so much have we made a drug, it's have we inspired others to do that. And there's a great example of work that my then still Dana-Farber colleague, Nathaniel Gray, and I did early on, in the very early days, where we were working actually with a pharmaceutical company on trying to make degraders of KRAS. And this was a 3-year project, involved a lot of people and a lot of resources. It failed at that point. We didn't get where we wanted to be, but we said, we did do the groundwork, and it turns out this pharmaceutical company, along with some others, went on, and now we have clinical trials of some of these molecules in the clinic. And this is really where I think we make the greatest impact, that we take this initial step and we really try to show people the way, and at a stage where it's too risky for any larger company to take on. And we've worked on principles, and again, this is very basic work. So in this case, one of the things we did, we really tried to understand mechanistically and structurally how do these proteins interact, what are the rules, what are some of the principles there that govern this. And we published this, and people pick it up, and I have a lot of other groups reaching out to me asking about this, so I know it's been taken up by the community. And again, doing this enables a lot of, in this case, a lot of biotech companies out there working with these principles, and within the time frame of 5 or 6 years, this field has really exploded, and there are now, I think, in excess of 30 clinical trials with new degrader compounds in clinical development. This is kind of a representation where they are, and I have to say that a lot of those are not yet where I would really like them to be. They're not in novel targets, they're not in novel biology, because it's, again, there's this de-risking, and it takes more, having been within a larger pharma company for a little bit, to convince something to take something on with a lot of biological risk, a lot of modality risk. So you see people go into very established targets. And so one thing we've been thinking about is, can we again help? Can we precedent this? Can we go into new indications, new targets, and maybe we won't have the resources to go all the way, but can we show people the way? And this is another example. We've taken on one of the transcription factors, or one of these piles of spaghetti, in this case a transcription factor called Helios, which was discovered actually at Dana-Farber to stabilize the phenotype of regulatory T cells. And the hypothesis was that if you degrade Helios, you would unleash the immune system to attack the cancer. And it was impossible to do this with any other technology. So we worked again with Nathaniel Gray back then and made prototype molecules, leveraging the same system I've just shown you is in play with thalidomide, and actually leveraging the fact that these molecules really like transcription factors. And we thought if we would modify them and if we would make libraries, we might be able to target other transcription factors. And it turns out that's the case. So we were able to make, in this case, prototype molecules that worked in cells and worked in mice, but wouldn't work in humans, but demonstrated that you can target Helios, the transcription factor. And now there are clinical trials from Novartis that run with a molecule very similar to what we did, targeting Helios, and are currently in clinical exploration. I think what I've shown you is just some high-level vignettes, and this is just one example of new technologies in therapeutic sciences, and we really try to work as a team here. And we work as a team within Dana-Farber, but we also work broadly with the industry out there. There's several companies that came out of Dana-Farber that try to take on these technologies and take them the next step. We've done very innovative partnerships, such as the Center for Protein Degradation. We're constantly looking, how do we broaden our impact? How do we take things further? I should also say that I'm really passionate about the chemical biology program at Dana-Farber being really a team play, where we have currently 10 faculty within the chemical biology program. And one of the missions we see for ourselves is we partner broadly across the institute and outside of the institute and really try to bring together all the expertise from the clinical side, from the deep understanding of cancer biology to our chemical mindset to think about advancing therapeutic sciences and advancing novel therapies. I think I might even be a minute ahead of time here. So just left to thank a great team, great lab, many, many people, and obviously thank all of you for your support of what we do here and everything you do for Dana-Farber. And I introduce Kathy Wu to join me up here.
Great, wonderful. It's wonderful to be here. I think, so my name is Kathy Wu. I'm the chief of the Division of Stem Cell Transplant and Cellular Therapies at Dana-Farber and the Lavine Chair for Preventative Cancer Therapies at Dana-Farber. And it's really a pleasure to be here. I think I'm going to be talking about something completely different than what Eric Fischer mentioned, but I think you'll see similar themes, namely the impact of advances in technology, what that has afforded us therapeutically. Also the theme of kind of our deep dive work to shine the path, to kind of inspire and motivate the field and try to transform the way that we think about therapeutics, and also the way that we kind of work together as teams. I think these are just kind of general themes despite the differences in subject matter that you'll hear. So now for something completely different, I'm going to talk about the immune system. So I think many of you are very familiar with what this system does. I will just briefly summarize to say that this is our army of defense that distinguishes between self and non-self. This is our first line of defense between external pathogens and invaders, and it involves every single organ in our body. There are many different types of immune cells that are key to coordinating together a strong immune response, but first and foremost, I will mention that T cells play a front row job in this whole system. And in terms of what are those external pathogens, our immune system is hardwired to recognize bacteria, fungi, viruses, parasites. These are kind of, they come in all shapes and sizes and flavors, and our immune system is really hardwired to deal with this heterogeneity and diversity. And a critical function of what T cells do is that they search and destroy. So in this kind of image on the bottom left is a T cell that encounters a cancer cell, and you can see successively as you go to the right, it kind of recognizes and goes to town, destroys. And this kind of critical function of T cells is really predicated on recognizing peptides that are on the surface of a cancer cell, which we call antigens. So these are kind of the recognition units that the T cells see. So how do we, it's long been a hope that we could mobilize this very essential function of T cells to help eradicate cancer. But first, just a little bit more about kind of a core functions of what T cells do. So again, they check for pathogens and mount a response against them. Our immune cells are constantly surveying in our skin, in our nose, in our mucosal surfaces, and if they encounter a pathogen, they start to move and they go to the lymph nodes and they start to coordinate in a coordinated response the generation of more cells and antibodies so that we can address that infection. And the other critical function of what T cells do is that they create memory. So they remember what was previously encountered. So what this graph shows is that if you have a first infection, yep, it takes a bit of time to mount a response, but if you come against that same the set of peptides, those antigens from those viruses or bacteria or whatnot, then the response is very fast and of greater amplitude. So this is what we call immunologic memory. And we've seen examples of these type of core functions at play over and over again. So this is an example that I like to use, which is a count that occurred on the Faroe Islands, which is up by Scandinavia. There was a measles outbreak back in 1781. Then there was no measles for 65 years, but then there was another outbreak in 1846. Those infected in 1781 were not infected again. So this gives you, were not infected again in the second outbreak in 1846. So this tells you that there is long-lasting immunologic protection if you gain that immunologic memory. Now, in more recent memory, for better or for worse, we know about COVID. And I think we will agree that the vaccines are basically what helped get us out of the lockdowns and all the trouble that we were in a number of years ago. And this was this idea that if we can give prophylactic vaccines, will that help us generate the antibodies or T cells that we need to protect us from subsequent infection? So now what about cancer? Well, cancer, for better or for worse, we are often talking about the situation of therapeutic vaccines. So these are our patients already have cancer. How do we mount a T cell response? How can we increase the breadth and the diversity of those tumor-recognizing T cells so that we can actually eliminate cancer? That's different than the infectious disease setting where we're trying to prevent. Now, for cancer, we hope to get to prevention. It's just that we still need some time to get there. For now, we're really dealing with therapeutic vaccines, but I do think that there are instances that that we hope that we can get to prevention, and Dr. Glimcher mentioned some of the different centers that we're generating so that we can start to think of preventative strategies. I do think that the rapid advances that we made in the COVID vaccine were helped by the decades of work that was already underway in cancer vaccines, and that helped feed forward and pay it forward to the rapid deployment of the COVID vaccines. But I do hope that now we're at a time that we can take the lessons learned from the pandemic so we can really make an impact on our cancer vaccines. Now, and this overall, I would say, just in terms of cancer vaccines, our general schema of how we think about how cancer vaccines could help us is that if the vaccines are there to expand and broaden the repertoire, could we not partner that together with immune checkpoint blockades like Keytruda, other agents that are currently being used in the clinic that can help release the brakes? There's adjuvants that we can use to rapidly accelerate the response, but can we not use the vaccines to help steer the response to recognize those antigens in the direction of anti-tumor, where we're not actually impacting normal tissue, so low toxicity, but high efficacy to actually get us closer to cancer cure. Cancer vaccines is not a new concept. It's been around the block. So what has changed? What's different now? And this is what Eric had alluded to, new technological innovations that have really given us the leaps to move forward. And I still remember this, a decade ago, when these two articles came out in the New England Journal back to back, and this was some of the early clinical trials that told us that immune checkpoint blockade antibodies could really create dramatic clinical responses for solid tumors. We have in the audience one of my laboratory neighbors and colleagues, Dr. Gordon Friedman, who was one of the initial discoverers of this really critical axis. But I think it's just been incredible how basic discoveries about the circuitry and the wiring of the immune system has led to such a huge impact in our field, such that cancer immunotherapy is another pillar of therapy. And I'd like to kind of akin the discovery of these immune checkpoint blockades to I call it the Obi-Wan Kenobi moment, where kind of, that goes, move, and suddenly the shield kind of goes down. So suddenly this vulnerability was there for cancer that allowed us to kind of move in. And to me, this is a little bit what the immune checkpoint blockade antibodies have done. The second huge innovation, also about 10 years ago plus, is the availability of relatively affordable and high throughput DNA sequencing. This is a graph that is very well storied among academic communities that study genomics, where large efforts such as the Human Genome Project and the Cancer Genome Projects have kind of sequenced all of these different tumors and try to look at kind of the mutation rate and burden across cancers. And what I would say that for me, this is a little bit like, wow, okay, so now that we can do sequencing, is this going to give us an opportunity to find those antigen, find those targets so that the shield is down, let's kind of go in. And so in the same way that sequencing has been really transformative in revealing to us on a population basis, but also on an individual basis, those mutations that drive those tumors, it has also opened up a therapeutic opportunity, because some of those mutations, if you kind of go from left to right, as the DNA mutations have the potential to change the sequence that then create amino acids. So the peptides begin to change. Some of those peptides will have the ability to bind to what we call HLA. This is kind of the ticket to get to the surface of the tumor cell. And once those peptides are on the surface, there's the potential for T cells to recognize them. So I mentioned before the antigen. So now you have tumor-specific antigens that the T cells can go over. And so I like to call these antigens, we call them neoantigens, because they're not in normal cells. And I would say they kind of hit the sweet spot, because by virtue of coming from the mutations are part and parcel of a cancer cell, they are very tumor specific. And at the same time, because they're recent events, they're coming from the cancer, the body has not been trained to dampen down immune responses against them. So they're highly immunogenic in the same way that a bug, that a bacteria, that a virus would be. So that's the sweet spot, this upper top right. And this is really a different place than where the field had been before sequencing became available, which is kind of in that big gray area where the field was really thinking about expression and shared across individuals as a way to target. So now with sequencing, suddenly there is this concept that, oh, neoantigens that have been theoretically thought to be interesting, well, now we can build computational algorithms that allow us from the sequencing data to integrate that with not only DNA but RNA information, but we also have machine learning algorithms that have been trained to allow us to predict what are those peptides that could mount a T cell response. So you put all that information together and out you go. You have a set of antigen sequences that are predicted to be highly immunogenic per individual. And this is also an important concept, because what we have known and learned from our technological innovation of sequencing is that cancer is not the same from one individual to individual. In fact, what we've learned is that each individual has a quite unique and different tumor. And so how do you deal with that heterogeneity? It's not one size fits all. Can we now, with sequencing, now that we know the set of neoantigens per individual, can we then specifically target that individual cancer, get to the cancer, don't touch normal tissue, and are we on our way to developing better immunotherapies? So at the outset, our group was really focused on building kind of the computational pipelines that would allow us to make these predictions. And our next question is, can we actually translate this to our patients? Can we actually build personal cancer vaccines and give them safely and efficaciously to our patient? And our group was among the first worldwide to generate a proof of concept clinical trial in melanoma. This could have really only happened here, where we had such a deep infrastructure for generating investigator-initiated clinical trials. We had the surgeons, we had the way to collect samples, we had the way to bring it into our cell processing lab, we had the way to predict those peptides and then to bring it back to our patients. And that the concept is shown here. We in real time sequenced our patient tumors. From those predictions, we were able to generate peptides that we pooled into 4 different pools, and we gave them as injections to our patients. And we gave them in what we call a prime boost schedule. We were trying to mimic what we think happens in a viral infection. So lots of early antigenic exposure up front with the prime, and then 2 boosts to stimulate immunologic memory. And some of our results were really immensely gratifying. We had 2 patients here, patient 2 and patient 6, who both had recurrent disease. But if you think back to that cartoon schema that I showed you, where we thought about, can we give the vaccine together with that immune checkpoint blockade? In fact, that's what happened with these 2 patients. They got the vaccine, then they got anti-PD-1 treatment, and within 12 months, 12 weeks, both patients had all of those lesions melt away, and we are now 4 or 5 years out later without any disease nor on any therapy. So certainly this has been a signal of hope for us to kind of, and positivity to kind of continue on this path. We've also had the opportunity to work with David Reardon in glioblastoma. I don't think for this audience I need to go into what a challenging setting that is. In our field, we call it a cold tumor, meaning like as opposed to hot and inflamed, where T cells are really not getting up there. So our question was, could our vaccine actually help get T cells, our soldiers, to where it's needed? And so we devised a clinical trial here, only possible at Dana-Farber because of our incredible infrastructure, where at the time of surgical resection, that gave us the tumor cells to go and sequence and then to predict and then manufacture and bring together a personalized vaccine that we gave when the patients finished their radiotherapy. And I'm happy to say that we were able, the colors here show you in the yellow and the green, those are 2 different flavors of T cells, and certainly before and after the vaccine, we can see increased numbers of T cells that we're able to get into this otherwise cold tumor. So I want to say in the last couple slides, I wanted to kind of show a little bit of how do we think forward in terms of how do we design our program. And it is really kind of an integrated program of discovery, but also feeding forward to clinical trials and back again. And so I've told you a little bit of 2 of our trials. We are focused on target discovery. We are focused on vaccine formulation and administration. And it is critical for us to understand the impact of the work we do. So we also do a lot of immune assessment. And so this has, this sort of kind of paradigm has allowed us to really, on the way left side, focus on disease-specific research. We have vaccine trials across many different tumors, and we do welcome the opportunity to get in deeper and understand better those malignancies so that we can do better with our vaccine strategies. I'm going to tell you briefly about our ongoing trials, and on the right-hand side, our approach to building new capabilities so we can maximize what we learn from our patients. And then, of course, taking all that information and feeding it forward again to think about the next iteration of trials. So first, what do we do in terms of our disease-specific research? Well, we really have been taking deep dives in the nature of those tumor-reactive T cells. These are, for better or for worse, relatively rare cells, so we have to have really great technologies that allow us to evaluate cells one by one. We are at a very interesting and exciting age of technological innovation, where now we are able to do single cell analysis. And way on the left, these sort of single cell analyses allow us to look into the phenotype of the cells. They also allow us to look at, for T cells, they express what we call the T cell receptor. It ends up being kind of a barcode, so we can actually figure out how one T cell or what we call a clone, is related to another. We can look at the hierarchy of the response. And finally, we want to know, what are those T cells seeing? How is this all related? And if we can understand that, this certainly will lead us to understanding new targets, new vulnerabilities, new approaches. Second, what about our trials? As I mentioned, we have a very exciting portfolio of investigator-initiated trials across the different disease centers in melanoma, led by Patrick Ott, glioblastoma by David Reardon, renal cell carcinoma with Tony Chuari, ovarian with Panos Konstantinopoulos, in CLL with, in the blood malignancies with Inye An and Eric Jacobson. So I've been so fortunate to be able to partner with my clinical colleagues and really devise smart trials that ask different questions depending on those different disease settings. And I just want to have a teaser. This is a recent trial we just completed in renal cell carcinoma. And what's exciting about this trial is, as routine patients get surgery, but then they get the vaccine. And what we've observed now, already 40 months out, is that no one has recurred. That is super exciting because this is a disease where, especially those with high risk, we do worry about relapse. In the right-hand curve, we did see one step down, and for better, unfortunately, this was due to mental illness related to COVID, but it was not related to disease. So I would say in the other patients, and so far we've treated 14, we have seen no recurrences, and we're really excited to see this type of approach go forward. In the last kind of bucket is kind of how do we think about new directions in kind of learning the most from our patients. And insofar as kind of understanding on an individual cell-by-cell basis, we're already starting to look spatially. So how are all these cells organized? How is their architecture in space? And we've been blessed to work with colleagues over at the Broad Institute in a technology which we call SlideSeq that allows us to get a tissue section on a slide, and then we're able to kind of computationally reconstruct kind of the architecture and look at the RNA level, the DNA level, and even the T cells and their clones. And so in my final slide, 2 slides, I'd just say, how are we getting to next vax? And this gets back to kind of my comments about COVID, which we've learned that, how can we get there faster and at lower cost, and how do we expand the repertoire of antigen targets that we get to? I think that's where the next level of innovation is required. We are hard at work because faster time and lower cost will mean more patients that we can treat, and more patients that we can treat means that we can answer larger questions related to prevention. So I want to thank you for your attention. I think that we've really thought a lot about tumor antigens. How do they relate to other cells in the tumor milieu? That we are really thinking hard about the lessons we've learned from the pandemic so that we can get to efficacious, cost-effective vaccines. And there's a lot of exciting data, not only from us, but in the field about the promise of personalized cancer vaccines. And I'm happy to talk to any of you about this. And again, what Eric mentioned, we are so grateful for our Dana-Farber family, the teams that we work with, and I thank you for your attention and your support. I think I'd like to ask Eric to join us, and we're certainly open for some questions.
Mildly probably confused, maybe expected. When you talk about vaccines, you're trying to activate the person's own T cells versus transplant, where we sometimes got in a transplant, and I'm one of the transplant people, where we receive T cells from a donor versus vaccines trying to activate. It seemed like trying to activate your own T cells. So
Yes.
I'm just repeating because
So you're trying to figure out who when we vaccinate, who are we kind of pulling to work? And I would say that in patients without transplant, yes, it is their own the patient's own T cells. In patients who have gone through transplant, it could be the donor cells. So we actually, I started my career thinking about vaccines after allogeneic stem cell transplant, and a lot of those principles helped me think through what we now have in our kind of non-transplant vaccine program. But vaccines can be given in many different settings, and because they're given as an injection, they will stimulate T cells, whether they're coming endogenously or from allogeneic and grafted source. We had another question? Yeah.
I haven't taken biology since 9th grade, so, but the question that I have is that my late husband had a mutation, and the way it was described to us was, yes, we have a targeted therapy right now, but the tumor is going to get smart, and it's going to outgrow, and it's going to get smart again. So I guess the question I have is that with all this innovation, all this research, all of the work that you're doing, these tumors are always going to be smart? I mean, is it a never-ending kind of, I mean, I'm being simplistic about it, but for a lay person, how do you make them stupid, I guess?
I would say, and you can chime in, if we only have one targeted therapy, the tumor is always going to get smart. The way to get there, and we actually learn a lot from infectious disease, is to have multiple targeted agents that are non-overlapping. So you're not targeting just one mutation, but you at the same time targeting another vulnerability of the tumor, and then maybe you activate the immune system. And once you have, I think, 3 to 4 threats to the tumor, it's going to be really hard for the tumor to outsmart you. But unfortunately, in some cases we are there, but in many cases we aren't there yet.
And this is not chemotherapy?
No.
Yeah, and I would add to that, which is that I think, what you're describing and others have described is like a game of whack-a-mole, right? So like you go, so I think it is a vote in confidence of synergistic combinations. And I would say that this kind of concept has informed us for how we design our vaccines, because we're not going against one antigen. We're trying to come in with 10 or 20. This is where cancer is also, a cancer vaccine is different than a COVID vaccine, because although there are some principles there that are similar. So I think, so not only kind of a complex vaccine, but also, I also mentioned combining it together with immune checkpoint blockade, like the Keytrudas, and even combining together with a targeted inhibitor, so that can actually shrink the amount of tumor that, so I think combinations are really what we're all thinking about.
This is really for both of you. Dr. Wu, you said during your presentation that this research could only happen at a place like Dana-Farber. And Dr. Fischer, you mentioned, I believe, that you had come from industry to Dana-Farber. Could each of you expand on what makes the research environment at Dana-Farber so unique as opposed to industry, or as opposed to collaboration across institutions or other cancer research hospitals?
Thank you for that question, and I'll start and Eric will follow, because I think we have different kind of experiences. But I would say, one of the things that is so special about Dana-Farber, and I kind of came through the system, I was trained through the system and stayed, is that I think there is a seamless kind of handshake between the clinical and the basic science. So what that means is that kind of the innovations can be born, but they can be implemented in a clinically informed and appropriate manner. And what that also means is that it's not easy to actually, we're unique, across different academic centers. It is hard to build the infrastructure that really makes beautiful, deep dive work possible. So what I'll say is the infrastructure to collect samples. So I don't know if Ted Pasquarello is here, but the Pasquarello Tissue Bank has been kind of really transformative for the way that we do our translational research. I think that having the cell therapy lab has also been incredible to support kind of actionable cell therapy, bringing it it to the clinic. I think that the ready access to sequencing resources are innovative ways that we can apply to our patient samples so that we can understand is really imperative. And then the other thing is the depth of kind of expertise across all the different fields in our midst, in our environment, has really been incredible to make sure that we're going in the right direction and we're doing it in the best possible way.
Yeah, I think I have to give you two answers. One is personal, my personal preference. I think there's a lot of things the industry does really well, and we work with them on many levels, so I think that's to be stated. But I really think this innovation needs to happen, and it's something I believe my passion is, and that is hard in an industry environment often because there are much more constraints. There's always a commercial aspect, and often these innovations happen in small indications. They happen in something that's clearly defined, which may not be commercially viable. And so it's easier in an environment like Dana-Farber. And then I think the second point I want to make is what struck me and still strikes me every day at Dana-Farber is how much all of Dana-Farber is purpose and less politics. Having seen the industry environment, they're really great people, but there's a lot of structure and there's a lot of politics or things that need to happen that aren't necessarily following just the purpose of doing something and getting something done. And what's been amazing in coming to Dana-Farber is how you can assemble. I work with people, and especially in the first couple of years, who may have not seen before. They're my colleagues, but it's a big place, and you're immediately on the topic. You immediately get people together, excited around an idea, and there's no, why should I do that? Is this going to help my division, or is it going to help a different division? There's a great idea. Let's run with it. Let's do it, and we'll figure out how to get there. And that's just, I think, something that makes Dana-Farber unique, probably also across academic centers. One more? Or?
Do you think you can combine your approaches with early detection when the tumor load is very, very low and maybe there's not as much diversity?
We always do better when tumor burden is low. So I think as a field we are evaluating how to bring together targeted approaches, immunologic approaches. The immune system is really great at mop-up function. So if you can get the tumor burden really low and do mop-up, that's going to give all that memory and long-term benefit. That sounds pretty good. So I think that certainly I think we've kind of given you two different, very different approaches, but really with the same goal that is actually quite synergistic. Thank you for your question. Okay. Thank you.